I'd like to introduce Abby. Abigail is our guest presenter for tonight's program. I'm just going to read because I don't want to get it wrong. She pursued her PhD as a student in the chemical biology program at UC Berkeley in the laboratory of Professor Matthew Francis. Abigail is currently working as a postdoctoral fellow in the laboratory of Professor Craig Hawker at UC Santa Barbara, researching polymeric materials within unique architectures and both biological and materials applications. So help me welcome Abby.
Awesome. Thanks so much for that introduction. It's awesome to be here. Thank you so much for the invitation. I'm super excited to tell you about my research cleaning up hexavalent chromium, which, as we now know, is a problem. So just to reiterate a little bit about who I am, I grew up in Charlottesville, Virginia, which has been big in the media recently. And then I moved to a very similar college town of Chapel Hill, North Carolina, where I did my undergraduate education. Then I moved to California to do my PhD at UC Berkeley. And this is where I did all the research that I'm going to be telling you about today. But for the past couple of years, I've lived in Santa Barbara, California, working in the Materials Research Institute there. All right, so sorry, I have a picture of Julia Roberts, but hexavalent chromium is a huge contamination problem. So this is just a couple examples of worldwide chromium contamination. So there are examples obviously in California, but it can be a much more significant problem in developing nations, especially like India. Here you can see the water is literally yellow from chromium contamination. And to my knowledge, you would never have seen that in California. With the contamination from PG&E almost 60 years, or over 60 years ago now. So the Environmental Protection Agency has limits for all of these different heavy metal contaminants, and there are a whole bunch that can be a problem. And today we're focusing on chromium, but all of these, if they are at levels that are above these regulated limits, can cause health defects in the surrounding populations and are incredibly challenging to get rid of. So the first thing I wanted to start with is what's in water besides water. So the water that you get out of the tap and the water that's in this pitcher isn't just molecules of water, right? There's a whole bunch of different ions. So there are negatively and positively charged molecules that are in this water additionally. And some of these things you might be familiar with. They might have already been covered in the Everything Matters series. So there's sodium and chlorine, and these form table salt, which you're probably familiar with. But additionally, there's some other ions. So this is magnesium and calcium. And there's not a lot in your water here in San Francisco, but there's a ton in Santa Barbara. So when I wash my dishes in Santa Barbara, there's this white film that I cannot get rid of. You can use vinegar and things to get rid of it. But that's because we have high levels of these magnesium and calcium ions in our water, and you fortunately don't. But there's also, so there's both positively charged ions and negatively charged ions that are commonly in water, and these aren't dangerous. But hexavalent chromium, which exists as this molecule with a bunch of oxygens bound to your chromium and then 2 negative charges, that is particularly toxic. And so something that you might be familiar with that can get rid of these toxic ions is this Brita pitcher. So I have a Brita pitcher for getting rid of the magnesium and calcium that are in my Santa Barbara water. And the way that this works is you have a whole bunch of ions in your water, and they're all floating around, and you have, and again, many more of these ions that are common and non-toxic in water than you would have some sort of heavy metal contaminant. And if you look at what's actually in your Brita filter, which you just buy and replace whenever the company tells you to, there's some activated carbon, which now I think, especially in San Francisco, they'll sell you in smoothies and stuff. But what this does is it pretty much just binds to everything. So this activated carbon will be particularly good at getting rid of things that are organic, things like bacteria. But specifically for the ions, there's an ion exchange resin. And so this is basically some sort of like plastic bead that has a charge on it. And so it's initially stuck to ions like chlorine and sodium, which are fine to have in water. And then when you expose it to a mixture of different ions, the ones with the higher charges will stick to your ion exchange resin, and then you'll release this chlorine and sodium into the water, which will make the water taste better and be safer if you have some sort of contaminant in there. And so another strategy that is used on a larger scale, and if you have like a water purification system at work, is reverse osmosis. And so basically what happens here is you have some sort of filter, and you push the water across the filter, and the only thing that can go through is the water. So none of your ions, none of your bacteria, none of your viruses, anything else that's in there. And so these strategies will all help to remove any sort of heavy metal contaminants. But the problem is that you're also removing all of these other ions that are in water. And so that means you either need a lot of material in the case of your ion exchange resin, or you need high pressure if you're using something like reverse osmosis. And so this is going to require basically a lot of money. And the problem with that is that these systems don't get implemented. And so if you look at the chromium contamination in California, the main strategy that was used there was basically to give these people different sources of water. There isn't really a good way of actually getting rid of all of that chromium such that we can use the water again. It's just not being drunk by people anymore. And so what we wanted to do was to develop a strategy that could bind specifically to those heavy metal contaminants and not to the other ions that were in water, so that you wouldn't need as much material. And so if you kind of imagine what this would look like, you have some sort of solid support, and so that could be some plastic bead with these, purple squiggle magical materials that are going to bind to your the heavy metal contaminant of interest, but not bind to anything else in water that's not going to be toxic. And so there's a bunch of different things that exist that can bind, that can be our magical purple squiggle materials that can bind to metal ions. And so this is an example of a small organic molecule. This is ethylene diamine tetraacetic acid. And while you might not have heard of it, it's in your shampoo, it's in a whole bunch of different household items. And it binds to metal ions, and that's great. It'll bind to a toxic metal ion, cadmium here. The problem is, just like that ion exchange resin, it also binds to things like calcium and magnesium, which are going to be in much higher concentrations. So this isn't necessarily a great option for what we were looking for. So biology has made some amazing materials, as I'm sure you're all familiar, and this is an example of a protein that exists in a lot of different organisms. And so it actually binds specifically to heavy metals, so it's really good at pulling heavy metals out of water. And some work that was done in the Francis lab, where I worked in at Berkeley, we used this protein in a gel in order to make sensors for heavy metal contamination. And so we would have a gel, and it would be big, and it would get small in the presence of these metal ions. And so the logical thing could be to take this protein and use it to remove all of the heavy metals from water. The problem with that is it's hard to make a lot of protein. Biology is good at making proteins. We're okay at making proteins. This one is pretty hard to make. And additionally, these proteins aren't stable. So if you could make a whole bunch of protein, there's going to be things in the water that's going to chew up the protein, and it's not going to last very long. So this isn't necessarily the best strategy for being able to remove heavy metals from water, but it does provide interesting inspiration for something that could selectively bind to a heavy metal contaminant. And so if you look at what a protein is made out of, this is what the structure looks like. And what's important is that you have this group right here, which is different on each building block, each amino acid. And the functionality that's right here in your sort of chain of these different amino acids is what builds your protein structure. And so there's all sorts of proteins in the body, and they do all sorts of different things. And what those functions are is determined mostly by what these series of functional groups are here. So what we wanted to do was to make something that was similar to a protein that had similar functional groups. And to do that, we made peptoids. So peptoids sound very similar to peptides and are in fact very structurally similar. So you have similar groups along your backbone, but if you remember, this pink R used to be right here, and now it's right here. And so you can incorporate the way these are synthesized. You can incorporate a whole bunch of different functional groups, even more than you can have with proteins. And additionally, excitingly, they're resistant to enzymatic degradation. So they can't be chewed up by enzymes in water. And so they're an interesting material to try and solve this problem of binding to heavy metal ions. And excitingly, they were actually developed by Ron Zuckerman, who's in the audience. So if you have questions, you should really talk to him. They're pretty cool. All right, so biology is pretty good at evolving things. It takes a lot of time. It makes all of these really cool functional proteins, but I didn't want my PhD to last forever, and so that's not what I wanted to do. What I wanted to do instead was to make a whole bunch of different materials and to test and see which one of those was going to have the function that I wanted it to have. And that's a strategy usually referred to as combinatorial chemistry. And so this picture is showing you a whole bunch of different beads which represent totally different materials. And we were looking for something that would bind to our hexavalent chromium. And so if you sort of zoom in on what each of these beads are, again, you have this sort of plastic solid support, you have this purple squiggle, and so this purple squiggle is representing our structure that's going to bind to the metal of interest. And so if you look at one of these beads, there's a bunch of purple squiggles, so that represents the same structure on that bead. But if you have two different beads, they're going to have two different structures. And so this way, with this solution or mixture of beads right here, we can evaluate the binding of a whole bunch of different structures by looking at all of these different beads. And so again, what these purple squiggles are going to be made up of is actually a very small structure. So proteins are typically over 100 amino acids, but we were making something that looks a little bit more like the ethylene diamine tetraacetic acid that I showed at the beginning. It's a small molecule with a little bit more diverse functionality than those small molecules. And so what we did is we made structures that looked like this, and we made approximately 2,400 of them. So with each of these structures, these functional groups, again, that give it sort of the interesting functionality that we were looking for are shown here. And some of these are structures that you could find in proteins, and some of them aren't, because again, you can incorporate different things with these peptoids. What we were just looking for was diversity of structures, hoping that we would be able to pull out a structure with a functionality that we couldn't have rationally designed. And so once we made all of these, we needed a way of figuring out exactly which ones were capable of binding to the hexavalent chromium so that we could pull it back out of water. And so we developed a screen. And so what we wanted in our screening conditions was we wanted an excess of those ions that you normally find in water. So again, your sodium chloride, your magnesium sulfate, buffers, you have sort of a neutral pH, and our hexavalent chromium ion. And so we were looking for, again, a structure that will bind specifically to that ion without binding to all of these other ions that are going to be found in water. But the question is, well, how do we tell which ones are binding to the hexavalent chromium? And so to do this, we turned to my advisor's book that he had from the 1940s of different tests for their colorimetric tests for identifying metal ions. And so there's a whole bunch of structures that will interact with different metal ions to give you different colors so you can identify which one is there. And so this particular structure interacts with our hexavalent chromium that's in water to form this pink color, which is great because we could use the pink color to find structures that were binding to this chromium. And so we wanted to test this, and so this is just a solution of hexavalent chromium. It looks kind of like that water that you saw at the beginning. And so when you mix it with this dye, you get a dark pink color. But again, there are multiple different forms of chromium, and this trivalent chromium is actually, again, in your body. It's in your vitamins. You need it to survive. And so we didn't want to detect things that were binding to trivalent chromium because that's fine. We don't want to remove it. And so what you can see here is that you don't really get a color change with your trivalent chromium. And any color change you're seeing here is likely due to just some hexavalent chromium that was in our original sample. So now, instead of an artificial picture that I was showing before, this is an actual photograph from the screen that we did. And so you can see a whole bunch of different plastic beads, a whole bunch of different colors. And what's particularly exciting about this photograph is here you have this dark pink bead. And so this dark pink indicates that in that bead, you had trapped a whole bunch of that hexavalent chromium and were able to dye it with this dye. But also, what was exciting is you also see these totally transparent beads. And what that indicates is it's not just that plastic that we were building the structures on that's sticking to our dye or to our chromium. We're actually seeing some sort of diversity in the response. And so we identified the most dark pink beads, picked those out, and figured out what the structure was. And here I'm showing 4 of the structures we identified. And again, these are peptoid structures. And something that was really exciting about the structures we identified is that there's similarities between them. Because again, I can show you a photograph of some pink beads, but what does that really mean? But having all of these structures and having trends indicated that we were finding things that could potentially give us an idea of how to remove the hexavalent chromium from water. And so you can see this is sulfur. So this is called a thiol group, which is known to interact with hexavalent chromium, so that was good. We also found groups that have nitrogen, and these can be positively charged. And if you remember, the hexavalent chromium is negatively charged, and so that makes sense as something that would interact with that ion. And so we're excited to identify the structures, but again, our whole goal was to be able to pull the hexavalent chromium out of contaminated water. And so we wanted to be able to evaluate whether or not we could do that. And so instead of making just one bead with one structure on it, we made a whole bunch of beads with this particular structure, because this one worked the best. We took the beads with this structure and we incubated them with some simulated contaminated water. And so to simulate contamination, my advisor said that we needed an ocean water sample, so I immediately went to Ocean Beach in San Francisco and collected some water. There's also a creek that runs through the UC Berkeley campus, which we used as a natural water source. And then we simulated contamination, assuming there was no hexavalent chromium in these natural water sources, which I think is hopefully a pretty safe assumption. And so these concentrations that we looked at were comparable to concentrations that you would see at contaminated sites. So you could see a lower contamination level, but in the water where you could actually see the yellow color, that's around this higher contamination level. So then we took our plastic beads that had those sequences of interest on them, shook it up with the simulated contaminated water, and then measured the amount of chromium that was left in the water after we did that incubation. And what we were excited to find is we were able to remove the chromium from water. And so what I'm showing you here is 3 different samples. So this is phosphate buffer, which is just a buffer that we have in lab. The unfortunate thing about using actual, real, natural water samples is we have no idea what's in it. So we wanted something where we actually knew what was in our sample, and that's this phosphate buffer. We also use, again, water from the creek and water from the ocean. And so these were all our initial simulated contamination levels at 200 micromolar. And we're able to remove a significant amount of the chromium using those plastic beads that had been modified with our structure of interest. And so I know today we're focusing on hexavalent chromium, which is a significant problem, but the strategy that we developed could be used for targeting a whole bunch of different things on the periodic table. So I'm going to skip ahead to a couple other elements, if that's okay. So we also looked at cadmium, which is, again, a toxic metal. And here we looked at whether or not we could pull cadmium out of blood. So right now, if you go to the hospital with cadmium poisoning, there's pretty much nothing that can be done. So we wanted to make something that could bind to cadmium in an even more complicated mixture. So in your blood, you're going to have proteins. You're going to have tons more other ions than you would have in your ocean water or in your creek water. And so we were able to develop structures that could similarly pull cadmium out of the blood without removing other things. We also made sensors in collaboration with other groups at Berkeley. So we made something that was fluorescent, and then in the presence of mercury, again, another contaminant, it was not fluorescent anymore, and so you could identify contamination that way. We also looked at uranium, which we weren't actually looking at as some sort of contaminant, but uranium, there's apparently a lot of uranium in seawater, and that's very useful for nuclear power. And so we looked at structures that could pull that uranium out so that we could actually use it. And so there's work continuing even now in the Francis Lab looking at other ions for other applications in collaboration with places like Dow Chemical to see if this selective binding can be useful for other applications. So with that, I just want to highlight some of the people that I worked with. So this is my advisor, Professor Matthew Francis, who I worked with. And this is an undergraduate that I worked with named Effie, and she is now a graduate student at the University of Illinois at Urbana-Champaign. And these are some of the people I work with now, but I just wanted to highlight how collaborative all of the research I have done is, and how diverse the amount of funding we've gotten is, which has been amazing. So thank you to these sources for funding, and thank you to you guys so much for your attention. And I think I can take a couple questions.
Thank you,
That's okay.
Abby. How about a hand for Abby? Yay! I have a question real quickly.
Yeah.
Can you also use your beads to just detect if chromium is in the water?
Yeah, definitely. So you could use it if there are low concentrations that you couldn't see. And also, while that pink dye seems to work really great for my assay, it also interacts with other things. So it's not necessarily as specific as the beads that we made. So you could use it to pull it out and then use the dye to determine whether or not there's chromium in some sort of water source.
Cool. Anyone have a question? Oop, way in the back was the first hand.
Are you or is anyone else working on removing arsenic from drinking water? Because it's a fairly common contaminant in New Mexico.
Yeah, there are definitely people working on removing arsenic from water. It's not something I particularly worked with, but I think this strategy could also work for arsenic as a contaminant.
There you go.
If you were to use the beads to remove cadmium from blood, how would you get the beads out of the blood?
So what we imagined, which we never actually implemented, I used fake blood for all of my samples, was something sort of similar to dialysis, where you would basically just flow blood through something where you could remove the cadmium back in.
There's a question over here. So, oh, Melissa.
Would
Look at that.
that same process, is that how you imagine getting the beads out of the water, or the water through the beads? Then
Yeah,
it's
so
filtering through.
we imagine something similar to like a water softener or the Brita filter, where basically, yeah, you just flow it through. And you don't necessarily, the beads worked really well for our screen, but you wouldn't necessarily need to have your structures bound to beads. It could be bound to some sort of membrane or any other sort of support, that's not necessarily my area of expertise. But yeah, it should work just as well with those kinds of things.
Yeah, what industry or what chemical industry produces the hexavalent chromium in the first place?
Yeah, so as was mentioned before, it's used a lot in pigments and platings, like the chrome platings, and in the tanning industry. And I think it seems as though there's been a movement to use less of it because it is toxic, but there's a ton of it already in the environment. So it's definitely still a problem.
We have any other questions? Thank you, Abby, and thank everyone here for coming to Everything Matters Tales from the Periodic Table.